All right. So cranial nerves, I'm glad you're starting here because there are two ways that these can be asked on the exam. So I can ask them of you from like an inferior view of our brain. So something like this, where the models that we have through the online resource are going to be really good review. So if you go to your resources module in Canvas, and I already have it pulled up here, but this is that website we have been using. The most important view for cranial nerves is going to be this view of the brain. So we can see everything here. Um, one of the best tips is that everything is in numerical order, and we name anterior to posterior and then medial to lateral. That is something that students tend to mix up. So if you start up top, start with cranial nerve number 1. This is your olfactory, and you can count down 1, 2, 3 is actually hidden in here. So some of our models are not the greatest. Some of them are pretty old. So we're not necessarily going to see 3. But in this case, we do have a nice view of things like our trigeminal nerve, or we've got this 6, 7, 8 relationship. You can see that medial to lateral. We've got abducens, facial, and then acoustic. So I'm going to focus on the stuff that is easily identifiable. I know not everyone is going to have cranial nerve 3, which is super visible. So if you just focus on the things that pop out to you from this view, things like our olfactory nerve, optic nerve, or this 6, 7, 8 relationship, things like trigeminal, number 5, those are fair game. And those are the kinds of things I'll tend to ask from this view. It gets a little messier when we get down to the brainstem and the medulla oblongata. We have to remember that relationship with the accessory nerve number 11 and number 12. So actually, I'm going to try to find a better view with our model so you can see that here. So we have this numerical naming system 1 through 12. And the exception to that rule is cranial nerves 11 and 12. They actually flip spaces here. So you can see they're the only ones that are out of order. This is our hypoglossal nerve number 12, which shows up on the model. I think that's like 44 or something right here. And then 11 is going to be a little lower. So 11 is just inferior to that. So hopefully that gives you some direction for the kind of stuff that I could ask from this inferior view. And then the other thing people tend to forget about is the skull questions. So there are going to be a couple stations on the exam where you have a skull with a wire running through it. You saw some of that on the practice exam the Thursday before spring break, but this is a good way to study that. What we have here is the names of the bony landmarks, those openings on the left-hand side, and then all of the cranial nerves that they are associated with. So I would pay special attention to any of these openings where we have multiple cranial nerves. These tend to be really good multiple choice questions. So I might say, identify one of the cranial nerves coming out of this opening here, which is jugular foramen. This is also a good fill in the blank question where you don't have multiple choice answers, but instead I ask you to give me 2 cranial nerves that come out of the superior orbital fissure, for example. So pay attention to those hot spots. And then some of these relationships we talked about before, like our ROS foramen, rotundum, ovale, and spinosum, all of those are going to have relationships to cranial nerves. So R and then O are going to be associated with our trigeminal nerve. We have that second branch going through foramen rotundum, and then our third branch going through foramen ovale. So anything like that that I've emphasized in class is a really good lab exam question. Any other questions on cranial nerves?
No, and for the online resources, the brain models are similar to the ones that are going to be on the lab exam, correct?
Yes, they are exactly the same structures. I would not try to memorize the lettering. I'm not sure if that's the same, but
Yeah.
from this type of view, this is exactly what you should use to study.
Okay, sounds good.
Yeah. So we can talk a little about the brain if that is going to be helpful to you.
Yeah.
Okay. So brain, we've talked a lot about this mid-sagittal view, and that is how most questions of the brain will be asked. Pay special attention to the midbrain and then the hindbrain. So I'm not going to ask too many things about the folds of the brain from this kind of view, but you need to be really solid on things like the thalamus and all the structures around that. So as per usual, it's always really good to identify your anterior versus your posterior sides when you're looking at any model. So if you start up here, this is going to be anterior. One thing you can use to confirm that is we have this pituitary gland here coming off that stalk that always points anteriorly. And then all of our brainstem stuff, that is going to be more in the inferior and posterior brain. So this is our posterior side. So from there, you can identify your thalamus, hypothalamus, which is just underneath it. And then we had a couple structures kind of arcing around the thalamus. Things like our C-shaped structures, our corpus callosum. We had that cingulate gyrus, which is this first fold here, this first gyri, going all the way around around the corpus callosum. We had the fornix, which is arcing back over the thalamus, and we also have a couple glands in here. So it is a very busy area. Make sure you go through those PowerPoints and make a list of all of these gold structures. So I'll just use the cranial nerve PowerPoint as an example. So anything that is highlighted like this in gold, go ahead and write yourself an answer bank. So I can't give you guys a list of structures, but you can certainly make one yourself from all of these highlighted terms throughout the PowerPoints. And the same applies to when you're studying the brain. I think that's one of the best ways to study. So there will be a long list of structures just in this region. As you continue down, we get more into some of these connecting structures. So we have this white matter made of these axons coming off of neurons. That's where we get this lighter appearance. And then things like our peduncle here are connecting regions of the brain. So you can use that relationship, white versus gray matter, as well. We also can't forget about the ventricle system. So we had lateral ventricles, which are not seen from this view, but we can imagine that this little septum pellucidum here at R is like this dividing barrier between our lateral ventricles. As we move down here, we get into this cerebral aqueduct. So basically our cerebral spinal fluid is going to be produced in this region here, just kind of an imaginary space where we would have that lateral ventricle, and it drains down and makes its way into this cerebral aqueduct. Now, the main ventricle that is visible and able to be labeled is at D. This is that fourth ventricle. It's that last collecting point for our cerebral spinal fluid before it drains into the spinal cord. So this would be a good thing to remind yourself of, this system of drainage into the spinal cord. And then we have these structures here of the hindbrain. For whatever reason, students tend to mix up the pons, which is this anterior lump. They mix that up with the cerebellum. So again, make sure you know your anterior side of the brain from your posterior side, and then that will help you to identify things like the pons versus the cerebellum and the medulla oblongata. Last thing I'll say about the brain is you want to cover all your bases, so make sure you're comfortable with this external view as well. Most questions will be asked from either the brainstem perspective or that midbrain, that mid-sagittal cut, but I could ask you about things like the lateral fissure, or we do have those pre and post-central gyri. Usually they might be labeled, and depending on the brain I use on the exam, you might have a marker labeling that gyrus, the dividing sulcus between the pre and post central gyrus. So those are good things to look out for. And of course, as you scroll down in this resource here, you will see that we have all of these things labeled for us. So if you're not exactly sure which of these grooves here is the central sulcus, look it up and you can see, oh, it's actually this groove that connects all the way down to our lateral sulcus. So that's how I would use this resource. Of course, you want to start with these unlabeled models and learn as you go. Check your work, check your answers with the labeled ones. This might be a good segue into spinal cord. So how are you feeling about the spinal cord histology and all that cross-sectional stuff?
I feel all right about it.
You okay? Okay.
Yeah.
I won't spend too much time on it then. I would just
And
say
then,
make sure. Mhm.
sorry, for histology, you said it's gonna be PowerPoints and microscope, correct?
Correct.
Like for the questions, okay.
Most of them will be microscope questions. So we haven't had that much histology. So maybe let's talk about histology and come back to spinal cord if we need to.
Okay.
Um, a lot of the histology is going to come from the endocrine system. So we have looked at, we had a little muscle histology, a little bit of neuroanatomy. We had those two special types of neurons, plus the basic structure of a neuron with the soma and the dendrites and the axon. But a lot of it's going to come from sensory and endocrine. There are two-part questions throughout the exam. So if you get a microscope question, a really good way to ask two-part questions is to have one identification question. You get a slide with tissue on it. I give you some sort of hint. Tell me what that tissue is. And then that part two of that station might be identifying a defining feature of the histology slide. So that table we did last class with the attendance sheet going through all of these endocrine tissues and figuring out unique cells or unique things to look for there are going to help you. So consult those tables, go through. I would recommend using the Human Protein Atlas to study for endocrine system because they have some really good examples. So what you can do is go through thyroid, go through parathyroid gland, and figure out one distinct trait for each of these types of tissue. For thyroid, we talked a lot about these follicles. So if you zoom in here, we have this repeating pattern of follicles filled with colloid. So if you get a slide that looks like this, if you can recognize that we have follicles all over the thyroid tissue, this is your ticket to getting this question right. So that's the kind of stuff I would practice with histology, is taking a slide like this. Can you recognize what it is off the bat? Or perhaps I give you a clue like T3 and T4 are produced in this gland, something like that. From there, take it a step further and identify unique elements, follicles, colloid, the types of cells around the follicles versus scattered everywhere else in the tissue. So that kind of stuff is something you can expect on the exam.
Okay.
Parathyroid was a tricky one. Depending on the slide box you have, you may not have adipose tissue. So I was clear in class, look at the Human Protein Atlas, practice with this kind of slide, because I will find an example like this to use on the lab exam if I decide to use parathyroid gland. So that's how I would go through for endocrine. And then we had our sensory histology. So for this one, your Yale resource is going to be your friend. We have an entire lab just for sensory systems. So you'll have to scroll down quite a ways. Once you get past the pre-lab quiz, we get into slides. So these are how I would practice to prepare for the microscope section. The main thing I'm concerned about with sensory is the eye and then the inner ear. Those tend to be the trickiest for students. We do have virtual microscope slides as well, so maybe actually these would be better for us. Don't forget about taste buds or those corpuscles from the skin system, but I think eye and inner ear tend to be the trickiest for students. So when you first put your slide under a microscope, it'll look like this. We have a cross section of the eye. So once again, you want to orient yourself. This is going to be anterior. I can tell because we have a really prominent lens here, and you get kind of this bulging out of the eye at the site of the cornea. And then way in the back, we have the optic nerve, which is a pretty big nerve. So it is really distinct. Where it enters the eye is at that optic disc or the blind spot. So once you've established those anterior versus posterior views, I'm really concerned with your understanding of the retina. So I could ask you stuff from this view, lens, cornea, sclera, but we have eye models for that. So any macroscopic structure like that is probably going to be showing up as an eye model question and not microscope. What I will do is I'm going to focus in on these three tunics we talked about. We had the outermost fibrous tunic, which is like connective tissue. We had a middle vascular tunic, which is where we have this choroid layer, which is really pigmented. And then we had our innermost neural tunic, which is everything else here. It's quite thick. So our eye slide, our eye question might look a lot like this, where you have this really distinct pattern of the retina. Now, I've told you in class, I want you to be able to identify the retina and all those three tunics, but then the only additional layer in the retina that you need to know is the photoreceptive layer. So if we get really close here, maybe at 40x magnification, we still have the stripy layer of the retina, but now we start to see the photoreceptive layer here, which is that layer very close to the choroid, but this is where we find those rods and cones. So know your retina, know your photoreceptive layer and your choroid, all that good stuff, and you'll be good to go. Now the inner ear is the other tricky one. So actually, let's go to the virtual microscope. This is messy. You know, you put it under a slide and it's hard to tell what you're looking at. If you remember the shape of the cochlea, it's like a snail shell. So what we are seeing is a cross section where we've got all of these layers spiraling up higher and higher, and that is kind of what we are looking at here. For the lab exam, we are going to see a view a lot like this. So with the inner ear, with the cochlea, we have those chambers, as they're called. So we have a lot of different terms for openings, so in layers. So tunics was new, chambers is new as well. What we're looking at is a cross section with bony labyrinth. So we have like a tube within a tube. That outer tube is bony labyrinth. And we're looking at a cross section of that tube right now. That's why we've got these three divided chambers. And then the innermost chamber is part of that membranous labyrinth. It's a softer, fluid-filled, basically network of tubes. So that's where we get these three chambers from. But before you try to identify any of them, find the shrimp. That's what I say. Purely because I think this organ of Corti here looks like a shrimp. So I tell people, find the shrimp, find your organ of Corti, which is that main auditory receptor of the cochlea, and use that as your guideline. So we have the organ of Corti, and then we have another landmark here, which is our vestibular membrane. And you may have a couple different names depending on which resource you're looking at. Any of those are valid, but I just call it vestibular membrane because that's going to help me identify one of these chambers. So once you have these two landmarks, the guy in between is the scala media. Media means middle, so this is our middle chamber. Across the vestibular membrane, we have our scala vestibuli. And then by process of elimination, the last one here is scala tympani. So those are some tricky words. I would spend some time working on spelling them out, making sure you have them locked in. Sometimes people get tripped up by that Latin, scala, vestibuli, these weird words that we don't use anywhere else. So, but that is the main stuff to look for within that inner ear. Does that make sense so far?
Yeah.
Okay. All right, let's see. Do you feel pretty good about the rest of sensory histology?
Yeah, I do.
Okay.
You answered all the questions I had, so I'm gonna head out.
All right. Thanks for
Thank
stopping in.
you.
You're welcome.
Bye. Have a good weekend.
You as well. All right, we're going to continue with a brief review of muscle. And I'm going to give you guys some muscle tips for studying. So let me just scroll down here, make sure I've covered all of these things I wanted to. A lot of people had questions about model orientation, and that applies to muscle. So we're going to look at some of those different models and figure out how we can tell anatomical position from all these different orientations and why we need to do that before we identify muscles. So let me pull up my PowerPoint here. Appendicular and axial. So I'll be flipping back and forth between these PowerPoints. and Canvas. So I'm not sure if everyone has discovered this on Canvas yet, but we have images of all of our muscle models. So if you're trying to practice and you're trying to figure out how much information you can recall, I would highly recommend going to Canvas, scrolling down to labs 11 and 12, and flipping through some of these models. We have everything for head and neck. Kind of creepy looking, but you can see we have a superficial side and a deep side for muscles of the head and neck from all of these different views. So take some time to go through here. It's, this is not that kind of comfortable studying where you're just checking something off a list, but if you can go through these and force yourself to identify what muscles you can, and then check your work, that's going to give you a good idea of where you need to spend some studying. So if you go through the torso and you think, well, I can identify my pectoralis major, but I'm not really sure what this muscle is, or these abdominal muscles. I can't remember the 3 names for external abdominal oblique. If you can't do that, make a note of the areas you're struggling with. But in order to learn the material best, you need to start with figuring out what you do and don't know from unlabeled models, and then go through with PowerPoints and fill in what you don't know. So enough about that. Let's briefly go through axial musculature. As I've talked about in class, I don't spend a lot of time on axial musculature on the test. So we talked about a lot of muscles of facial expression. Some of these are visible on your model, some are not. So that does affect what I can ask you. Things like our orbicularis oculi and orbicularis oris are really nicely labeled. Those are the sphincter-like muscles around our eyelids and around our lips. So I would pay attention to those, whereas things like our buccinator, they're a little deeper. So buccinator, some of these muscles of mastication, like our masseter, we have to dig a little deeper to identify them. So I'm really going to keep it superficial. I hope that helps you with your studying, figuring out which of these muscles are most superficial. Things like the orbicularis muscles. We have a nice frontalis. The zygomaticus muscles tend to pop out on our models as well. So use those to direct your studying. As we move down into the neck, we spend a lot of time on sternocleidomastoid. Remember, the name comes from the attachment points of this muscle. So it has an attachment point at that mastoid process, and then down on the sternum and along the clavicles. So we spent so much time on this. I hope that gives you a good idea of whether you can expect to see it on the lab exam. But some of the things that I might use when I'm writing these kind of muscle questions is I will give you hints about the attachment points or the actions of the muscle. My questions will be purely identification. So you need to be able to identify a muscle based on where an arrow is placed on a model. So you don't need all of this extra information to do that, but as I write my questions, I might ask, what is the name of this muscle which bilaterally contracts to flex the head? So all you need to do is identify what the arrow is at. But if you know that the sternocleidomastoid bilaterally contracts and that causes head flexion, that is a really good clue that you are on the right track to identify this as sternocleidomastoid. So all of this is good information. That said, you don't need to know attachment points and actions to do well on the exam. As long as you can recognize these muscles on a model, you will be golden. Get a lot of questions about posterior and anterior triangles and strap muscles. So while we did talk about these guys, they're not going to be emphasized again because I don't want to have to go digging around on a model to find them. I just have 30 minutes to set up. So stick with the superficial muscles of the neck. Things like the borders of our triangles are good questions to ask. So we have the sternocleidomastoid, providing that border between the anterior triangle and the posterior triangle. We also have trapezius forming the back wall of that posterior triangle. If we go back, forward. Anterior triangle is where we get into all of these muscles, which are called the strap muscles. So these are much more testable in lecture. You can expect to see them in lecture, but I don't tend to emphasize them. We have a lot of muscles of the superficial trunk. So look at your fiber orientation on your model to help you identify things like our muscles of the abdominal wall. Remember, external obliques run like hands in your pockets. So they run inferiorly, kind of down and towards the belly button. Internal obliques do the opposite. They run superiorly in the opposite direction, and they are the second deepest layer here. So I'll show you in a second a model with a superficial side and a deep side so you can tell apart these layers. And then we have that deepest layer, which is transverse abdominis. These fibers are running transversely or horizontally. And of course, most people are familiar with the rectus abdominis. That is going to be on the superficial side of our model. But we've got this straight fiber orientation with tendinous insertions forming that six-pack. So in addition to the rectus abdominis, you do want to know tendinous insertions and linea alba, which is that white line along the midline of the abdominal wall. Again, these are much deeper, so I don't tend to ask the intercostals. Diaphragm is good to know for lecture, but that is not testable in here because we don't have a great model showing it. And then we get into kind of this overlapping region of superficial back and some of the upper extremity muscles. So I've gone through these in class and put arrows on the model. Hopefully you took photos of that in class or you know someone who did. But everything here that is highlighted in gold is good to test. Again, focus on superficial muscles. What can you see from a glance in the superficial back? We've got trapezius. Slightly deeper, we have the rhomboids here, which form this rhomboid kind of quadrilateral shape. Latissimus dorsi is this really broad muscle of the back, so it is easily labelable. It's easy to put a label on it. We have things like the deltoid as well, that cap of muscle along the shoulder. And then we get into these deeper muscles. So again, the really deep stuff like iliopsoas is not very testable on the exam. Okay, so I know I ran through that quickly, but we don't spend a lot of time on axial muscles on the exam. So let's go ahead. I'll show you some of these models and then we'll run through our appendicular musculature. Okay, I actually think we want to look at the torso. Let's see what our half head can show us. So this is our half head model. Very large photo. So as you can see here, a lot of those muscles of the head and the neck that were deeper, like the strap muscles or the posterior triangle, those are not super visible here. So we do see things like the temporalis. We do have a nice view of the masseter, that main muscle of chewing, frontalis up top. And then if we switch views here, whoa, let's get an anterior view of the head and neck. So back to our creepy guy. So again, we have things like frontalis, these orbicularis muscles make good questions, and then we do have our zygomaticus muscles, which are paired. So we have a zygomaticus major and minor, but they tend to get grouped together on the lab exam. So there's many less muscles that are viewable here than there are in the PowerPoint. So I hope that helps. Again, stick to the superficial stuff, and let's run through abdominal real quick. Same thing here. So we have on the left, well, on his right-hand side, we have this rectus sheath, which is all that connective tissue. It's basically taking these aponeuroses from all of our abdominals, and they run together towards the linea alba. So that is the most superficial view. If we go a little deeper, we see our rectus abdominis with those tendinous insertions. And then we also have a little bit of our external abdominal obliques. That's pretty much all we can see from this model. So we don't see internal abdominal obliques, we don't see transverse abdominis, and those things are a little too deep to be identifiable on this kind of torso model. All right, moving on through to appendicular muscles. This is where you're going to spend most of your time when you are studying for the muscle portion of the exam. I've talked a lot in class about how to study muscles. I would recommend going through these first slides again, just to remind yourself, try to visualize what actions are happening at a muscle. That can help you on the exam. Remember, if a muscle crosses a joint, it's going to act on that joint. So if I include any hints in my questions about muscle actions, ask yourself, I think if you know or suspect you have the right muscle, ask yourself, does this muscle cross the joint? Does it make sense that my answer aligns with the hints I'm given? So if you get a question asking you to identify biceps brachii, and I tell you this muscle flexes the arm and the forearm, or this muscle crosses the elbow and the shoulder, those kind of hints can help you if you are familiar with the actions of a muscle. Flashcards are okay, but I want you to group things by muscle groups rather than trying to make a flashcard for every single muscle. That is a great way to get overwhelmed by the material. So if you are going to make flashcards, what might be helpful is to go through your lab manual. We have these really nice tables with just so much information about muscles, probably a little too much for this class. But you can go through here and make flashcards for each muscle group, like the gluteal block, the lateral rotators of the hip. These are all grouped together already in your tables. And now you can take the most important information from there and try to identify patterns. So this is an example of how I mark up my tables when I'm trying to study for something. Try to identify landmarks that are common to multiple muscles, common attachment points, common actions. So when you're making flashcards, group these things together. Don't try to make a flashcard for each individual muscle. All right, again, focus on the superficial stuff. So we just talked about back, so I'm going to kind of gloss over that. I will show you this on a model, but we do have a superficial side of the back and a slightly deeper side on our models. So between these two, pay attention to things like trapezius, deltoid, latissimus dorsi, and on that deeper side, we do have muscles of the rotator cuff and then muscles of scapular retraction. So our main scapular retractors are going to be the rhomboids. Remember, minor is above major. So these guys are right in between the scapula. And then we have our rotator cuff muscles. So use the scapular spine as your landmark. If I put an arrow on the muscle and it's on the shoulder blade itself, and this arrow is below the scapular spine, then that tells you it has to be infraspinatus. If the arrow is above that scapular spine, then it has to be supraspinatus. Let's see. Yes. So serratus anterior, as I said, it's one of my favorite muscles. You can probably expect to see it on the exam. I remember that guy wraps around the ribs. So it's kind of this weird in-between muscle. It's not really part of the back, but it's not really part of the anterior abdominal torso region. But it does do an important role of protracting the scapula. So you can look for that on our small muscle man, the guy who's kind of lifting his arm and looking upwards. He's posing for us. But you see all of these serrated muscle fibers here weaving into our external abdominal obliques. All of this is going to be serratus anterior. Okay, covered rotator cuff. I think most people are more comfortable with chest and arms, but do remember we have a superficial muscle here, which is that fan-shaped pectoralis major. And then on the deeper side of the model, we have a much smaller pec minor. This guy's only attaching to ribs 3 through 5. So it's much smaller and it doesn't play as active as a role in arm actions. So make sure you're comfortable with those guys. And then we get into our compartments. So the main challenge with our models is figuring out anterior versus posterior, lateral versus medial, figuring out where anatomical position is. So I'm going to show you our arm model and then our thigh and leg model. These are all available in Canvas, and I recommend flipping through these guys as well. This is where we get into compartments. So the arm, forearm, thigh, and leg all have muscle compartments that share common actions. So again, if you're making flashcards, it would be a really good idea to make a flashcard for each muscular compartment. That guy is blurry. Cool. Okay. Do we have a photo for the whole thigh? That's a question. There we go. Okay. So we are in the thigh. This is our thigh and leg model. And when you look at this, you might not be sure which side is lateral versus medial. I would start up top. This is the iliac crest of our pelvis. So this is that lateral part of the pelvis, of the ilium specifically. It's arcing this way. And then all the way down here, we have our pubis. So this is our midline. This is medial, whereas that iliac crest is lateral. So if you are ever confused and you're looking at the thigh, find your pelvis and figure out medial versus lateral. From there, we know that this has to be our anterior compartment. So this is where we have muscles that are going to extend the knee, and we have one case that is going to flex the hip. So the main muscles of our anterior thigh are the quadriceps. These are very popular to ask on the exam because they are nice and superficial. So this is going to be that rectus femoris. It's straight up and down. This is that muscle of the quadriceps that crosses both the knee and the hip. So it is going to both flex the hip and extend the knee. Overall, all of these muscles are running into the quadriceps tendon, which goes over the knee and then forms the patellar tendon or patellar ligament. Either term is fine. We also have the sartorius, that S-shaped muscle that crosses from the lateral hip down to the medial knee. And that's what allows us to sit crisscross applesauce. Last thing we can see here would be a little bit of our TFL, our tensor fasciae lata, which is this muscle that blends into our glutes, and it also blends into our IT band, the iliotibial band, which runs all the way down to the lateral side of the knee. So this is another good way to identify lateral versus medial. The IT band is lateral, and then we have this teardrop-shaped part of our quadriceps, which is more medial. Let's compare that to the arm model. Arm is also tricky because it's not quite an anatomical position. So if you're looking at a muscle man, we have an arm that is extended and abducted. So it's not laying like it would in anatomical position. But what we can see is the palm. We see the thumb is lateral, so that is how we would expect to see it in anatomical position. And then the palm side is facing forward. So this tells us we have to be in the anterior compartment of the forearm. That's where we have our wrist and forearm flexors. We have the anterior compartment of the arm itself, which is mainly biceps. Again, that's the most prominent superficial muscle here. So I would say spend most of your time breaking down muscles of the forearm. That is the trickiest area, in my opinion, of all of the appendicular musculature. So just as with every other muscle compartment, the forearm is divided into muscles with common attachment points and common actions. So we do have multiple layers in each of these compartments, anterior versus posterior forearm, but focus on superficial. It's always going to be superficial in this class. I spent a lot of time talking about this PFPF memory cue. So let me find the slide here. So if you take your right forearm and extend it out, take your left hand such that your fingers are like so, and put that index finger so it is pointing towards your thumb. So it's pointing laterally. We get this PFPF relationship. So you can do this on an exam. You're not going to look weird. It's totally fine to act out this PFPF on yourself if you're trying to identify forearm muscles. So these knock out our superficial forearm muscles. Index finger is pronator teres. That is the most superior muscle that runs medial to lateral in the anterior forearm. Next is flexor carpi radialis. So this muscle is going to run from medial elbow all the way towards the radial side of the forearm. Palmaris longus is this weird muscle that has a tendon that goes down to the wrist and then jumps over flexor retinaculum. I'll talk about that in further detail in just a second. And then flexor carpi ulnaris is this last superficial muscle of the forearm, which runs down the ulnar side, or the pinky side of the forearm. So let me back up a couple slides so we can see those relationships. So this is PFPF. Don't let this confuse you though. We're looking at the left forearm now. So the left forearm has been extended, and basically we've taken our right fingers and laid them across the forearm. So we still see PFPF. That weird muscle I was talking about, palmaris longus, is this guy here. So you can see the tendon runs all the way down, and then instead of going underneath this band of connective tissue, which is called flexor retinaculum, instead of going under that, the tendon is jumping over that, and it inserts onto the palm. So that is another really handy way to identify this muscle. And on our model, it's a little hard to see here, but you can follow this tendon down and see how it goes over that band of connective tissue. So this right here is our palmaris longus. All right. So those are the main things to study for the anterior forearm. We'll go quickly through posterior forearm. And in this case, we only have these two layers, but they are a little busier. But again, you want to focus on that superficial layer. So posterior forearm is where we have our extensors of the forearm. So you're going to see a lot of names that have the term extensor in them, and then they're followed by whatever the muscle is extending. So for example, extensor digitorum is this muscle in yellow here. It runs all the way down, and you can see it splits off into tendons going to digits 2 through 5. So there's 4 tendons in total here. That's the case for all of these extensors. So we have one going to digits 2 through 5. Digiter minimi is going to just our pinky finger. And then we have extensor indicis, which is deeper. So in addition to our extensor digitorum, that is a really good candidate for a lab exam. I'll just tell you that upright. The things I'm looking for to test you on are muscles that have really distinct attachment points. So things like extensor digitorum or our PFPF muscles, those are very testable because you can clearly tell based on the tendon pattern that there are 4 tendons here going off to the digit, so this has to be our extensor digitorum. So I hope that helps as well. That is another reason why extensor carpi radialis longus and then extensor carpi ulnaris are also really good test questions. So if you break down this really annoyingly long Latin name, we're still extending the wrist. And in this case, this muscle is running along the radial side of the forearm. So if we go to our image here, find your radial side, your thumb side, and then we have this muscle in blue running all the way down that radial side of the forearm. Compare that to something like extensor carpi ulnaris. This is running down the ulnar side of the forearm. We can see this is our little finger here. So this muscle would be extensor carpi ulnaris. So I know the hardest part is remembering all of these really long Latin names, but if you can think about what action is happening and what is it acting on, that will help you remember these kinds of names instead of just trying to purely memorize. So all those things I just talked about are visible on our model. Let's go down here, posterior view. So now you can tell this is posterior view because we're seeing the back of the hand. We can also see the back of the shoulder. So the deltoid here is that cap of muscle on the shoulder. So both of these are clues that we're looking at the posterior arm and forearm. So again, follow the tendons here. Look for the patterns in the tendons. We have this muscle with tendons branching off. Let's count them. 1, 2, 3, and then we have a little bit going to 4. So this has to be extensor digitorum. You can do the same thing for the extensor carpi muscles. So figure out, are you on the radial side or are you on the ulnar side of the forearm? This is radial, so this has to be our extensor carpi radialis longus. So it's hard to see on the model, but this that I'm circling is that muscle. And then on the ulnar side, we have, again, another muscle here, extensor carpi ulnaris. All right. So that about covers everything I wanted to talk about with muscles. So notice I did not cover every single muscle that will be on the exam. We did gloss over thigh. We glossed over leg as well. So you still want to go through and make your list of terms and make sure you are comfortable identifying them. But think about the things I talked about in class. Superficial muscles are king. So spend your time accordingly. Okay, let's see. Openings at the skull. Okay, last thing we're going to cover is muscle histology and then the spinal cord. So let's start with muscle histology since we're already on the topic. Let's see. And I'm going to use the Human Protein Atlas to help us. So muscle, as we learned in Unit 1 and Unit 2, there are 3 types, and they're all of them pretty distinct. So today I want to focus on skeletal muscle. I feel like we've pretty much beat to death the cardiac muscle and the smooth muscle and the differences between those two. So you can refer back to those PowerPoints if you are struggling with them. But the newest stuff for us is skeletal muscle and then all of these smaller subunits. So when you get a slide, you'll have to zoom in quite a bit. And you start to see these really prominent striations. If you see a muscle slide that looks like this, where this kind of jumps out to us that we have striations all throughout the tissue, this has to be skeletal muscle. It's the most striated of all of them. If you see a pattern like this, it has to be that skeletal muscle. We can zoom in further now. So in our introduction to muscle histology, we talked about the basic contractile unit of muscle, which is the sarcomere. So in lecture, you probably talked about A band and I band, all of these different zones and the Z lines and that kind of stuff that make up the sarcomere. We're not going to be that detailed on the lab exam, mainly because I can't zoom in that closely with a microscope. So this is pretty much as far zoomed in as we can get with our 4DX microscopes. What we can see is that striation pattern. So I could put an arrow on one of these stripes here and ask what causes this pattern, or this pattern is caused by the basic contractile unit of skeletal muscle, something like that, where we have those hints, and what I'm hinting at is the sarcomere. So that is an additional structure to know for skeletal muscle. And then we also can look at a cross section of skeletal muscle, where instead of seeing those striations, we're looking basically down on a cut cross section. Trying to find a nice one for us. Maybe see what Yale has for us. So we can look at a cross section of muscle and see some of connective tissue structures. So again, I'm scrolling down to slides. Here we go. So this is our cross section of muscle. Rather than seeing striations and just really long muscle fibers, we now see the cross section of those fibers. So they look roundish-ish. And we see some of these connective tissue structures that divide our muscle. So remember, the biggest unit of muscle is the muscle itself. That is the entire muscle organ. Muscle can be subdivided into fascicles, which are bundles of muscle cells. And then we get to individual myofibers or muscle cells. And that's what we see here. So each of these roughly round, maybe triangular shapes is one muscle cell. We have a really tiny connective tissue membrane surrounding all these muscle cells. That is our endomysium. Endo means within. Surrounding each fascicle, we're going to have a perimysium, which is, peri means around. So this is surrounding every fascicle. And then the very thickest kind of connective tissue that you will see is going to be that epimysium. So probably not going to see that one at this level. Instead, we have, for sure, we have perimysium and endomysium in addition to all these muscle cells. So make sure you are covering both your cross sections and your longitudinal sections. Last but not least, we are going to look at a spinal cord. Now, there are two ways spinal cord questions can be asked, and that is from a model or from a histology slide. So the very first lab of neuroanatomy was spinal cord. Let me pull up our model, and then I'll also pull up some histology. Okay. Okay. Not loving that model. Do we have a better one? Yes, we do. Okay. So this is the model we'll use on the exam. We can see it's a lot nicer than the other one. Hasn't seen as many years. Okay. So spinal cord, in context, it sits within a vertebrae. So we're going to have this entire spinal cord running along the vertebrae within that foramen. So the way to orient yourself here is to look for the body of the vertebrae versus the spinous, that spinous projection is going to be more posterior. So once you find your body here, this tells you this is anterior. And then we have that spine posterior. Spinal cord sits in the middle here. So we talked about 3 layers, dura. So we had a dura mater, a pia mater, and an arachnoid mater. Not in that order. So the dura mater was the outermost protective layer. That is going to be not super visible in this model. So I'm kind of highlighting it here with my mouse. But this kind of grayish blue is going to be dura mater. The arachnoid mater is that middle layer. It's cobwebby. Again, not super visible on our model. Same with pia mater. So the main thing I might ask could be the dura mater, which is continuous with that outermost covering of our nerves. We also have an epidural space. So there is this fat-filled space here. You can see the yellow is made of adipose cells or adipocytes. This is that epidural space where if you get an epidural injection, they're going to stick the needle into this fatty layer. There's less chance of damaging the spinal cord because of that. So that is stuff around the spinal cord. And then we have the spinal cord itself. So remember, the organization of gray versus white matter is flip-flopped in the spinal cord. So this butterfly shape in the middle is our gray matter. We're going to have sets of horns. So again, has to be anterior because the body of the vertebrae is more anterior. So these first set of horns are going to be our anterior gray horns. They look like little bumps. We have lateral horns as well. So those are slightly more prominent bumps, but you can see they stick out laterally on either side of that butterfly shape. And then the posterior gray horns are the pointiest. So they are the most horn-like of these pairs of horns. We have a central canal in the middle, and then this part here, this connecting part, is the gray commissure. So that stuff's not too complicated. Just check your spelling, make sure you can spell commissure, and make sure you don't confuse gray versus white matter in the brain versus spinal cord. Other things to note would be our depressions here. So make sure you know anterior from posterior when you're identifying your fissure versus your sulci. And then we have some roots. So I think people are understanding the concept of ventral and dorsal roots pretty well, for what I've seen on the practice exam. But again, you always need to figure out your anterior from posterior view. Those correspond to ventral versus dorsal. And that is the case with the roots. So we have a ventral root with no ganglion versus a dorsal root, which is this guy coming off the dorsum or the posterior side of the spinal cord. And this does have a ganglion. So we have this dorsal root ganglion, which is really just a swelling of the root. It's where we have more neuron cell bodies. So that is only found on the dorsal root. That is another clue to tell your anterior slash ventral from your posterior or dorsal sides. Some of those things can also be asked from a microscope question. So our slides, most of them do not have the roots. So this is actually a really nice section. We have preserved the ventral root and the dorsal root ganglion. What you will most likely see on the lab exam is just the cross section here. It may be blue, it may be gold, it may be a different color. So don't let the color of your slide throw you off. But look for the butterfly shape. When you don't have things like the roots or the vertebrae to help you identify anterior versus posterior, you will always have the butterfly shape of the gray matter. So the pointy part is always going to be posterior gray horn, whereas the more rounded edges here are more anterior. We've got that anterior horn, and then the side bump is the lateral horn. So when you don't have all those other external features, use the horns to help you. Same thing with fissure versus sulcus. All right, and with that, we have covered all of the main concepts I wanted to get to today. So again, this is not an exhaustive list, but this should hopefully get you on the right track for what you should prioritize for the lab exam. If you have any questions before the lab exam on Thursday, bring them to class on Tuesday, or feel free to message me. I'm available through Canvas. Otherwise, we will keep trekking with our content on Tuesday, and good luck as you prepare for that lab exam.